Chapter 2, Section 7, Cell Inclusions. Now, it turns out that, yes, we have stated all along prokaryotes do not have intracellular membranes. There's no organelles. But it doesn't mean they're not storing stuff inside the cell, just like we use our ER for, many different purposes. One of those purposes happens to be the storage of calcium. Same with the Golgi, store stuff there. We have all these small vesicles, lysozymes and whatnot floating around in our cells. Well, bacteria do some of the same things. Archaea do some of the same things. We refer to these storage mechanisms as inclusions. Okay? They're not membrane bound so much, but they're going to be just this clump together of proteins, metals, or some form of blank. There may, a lot of times there's going to be some thin proteinaceous membrane, not a lipid membrane. There's just proteins wrapping around it, encasing it, holding it together. There's going to be ones that are carbon storage. They call them poly-B hydroxybutyric acid, PHB. These are going to be long chains of organic molecules that have been overly synthesized. And instead of breaking it down catabolically and then have to go through all the time and energy to anabolically replace these at a later point, take this overproduced stuff and clump it together somewhere in the cell. Here's a ball of PHB, a ball of PHA. You can break that down later and use what you need of it, leaving the rest there for another time. You can see here from figure 2.220. Here is, some of that PHA, that, butyric acid, these large, looks like globules of, boom. It's all of this just joined together there that the cells can break down and utilize as needed at other times because they've overproduced it, because whatever building blocks are there, while it's there, might as well just kick in the anabolic metabolism and just overproduce, store it, and because at some point in time, whatever that was in the environment is going to deplete it or it's going to go away. Same with many other much needed compounds in the environment. Phosphate, sulfur, carbonate minerals. These are going to be other things that are going to be found that are going to be packaged away and stored in different bacterial cell. You'll see polyphosphate granules, inorganic phosphate. Going to need inorganic phosphate for numerous things, nucleotides, some making of ATPs, replacing, replenishing, and all of this stuff. So if it happens to be there's a lot of that inorganic phosphate in the vicinity that's here today, gone tomorrow, take up as much as you can and just store it in granules, large conglomeration of. Same with sulfur. They call it periplasmic granules, where you just take a bunch of sulfur-containing compounds and just glom them together. Boom, 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 form almost this pseudo-crystal-like. Stored, it's there. You can break it down or break off little bits of it later and use what you need. Leave the rest stored. So you can see the polysulfate, these little black dots, they're showing, this, here they are, these granules inside the bacterial cells. Same with the sulfur. In these spheres within the cell, those are sulfur compound containing granules that have all been glommed together and stored. As needed, they'll be broken down. Same here, you see this biomineralization, taking up minerals that are going to be needed. some point in time in the future. Turns out there's a lot of it here and the now. Don't know about the future, so these cells have evolved to take it up now, store it away now, in case it's, goes away later. You can also see that there's going to be gas vesicles for some of these microbes. Gives these guys a level of buoyancy. When you look at the cyanobacteria are pigmented. They have a rudimentary photosynthetic properties to them. To be photosynthetic, or at least even pseudo-photosynthetic, they have to be up near the surface of the water where they can be hit by light. They sink down too far into the water column. Well, it turns out that UV radiation, those photons of light, aren't going to be able to pass down far enough for them to be excited. So they need to be up at the top. What they've evolved to have are little gas vesicles, make them buoyant, kind of like you when you wear a life preserver. You're always going to float to the top. Same with these guys. Turns out some bacteria respond to magnetic fields. Different magnetic fields will alter the way that they move around in the water column. They can refer to this as Magnetotaxis. Okay, the different magnetic fields. Well, for them to be able to respond to magnetic fields means they're going to have some form of biomineralization of magnetic iron oxides in them, stored away. That's what these little bricks here are inside. The pulling and pushing of the magnetic fields pulls and pushes these inclusions, which will trigger different responses from the cell.